Okay, first I would like to thank the scientific committee for the opportunity to share with you some thoughts on the design and implementation of clinical trials during a public health emergency. I will be drawing from experience gained by my involvement in some trials conducted during the current global pandemic of COVID-19. Okay, so here is an overview of what I will be covering in this talk. I shall very briefly summarize some of the lessons learned from previous pandemics, and in particular, the Ebola outbreak in West Africa in 2014-15. I will then give a high-level overview of adaptive platform trials with multi-arm, multi-stage designs, or MAMS for short, and I will focus on the application of these designs in therapeutic trials conducted in the current pandemic of COVID-19. Finally, I will touch on some of the issues in the design and analysis of trials of vaccines against COVID-19. Is posed by public health emergencies caused by a new infectious pathogen. There are no approved treatments and vaccines, and knowledge of the disease and understanding of the epidemiology are evolving. Yet, there is a need for developing therapeutic and preventive intervention at great pace. During the last Ebola pandemic, a number of therapeutic trials were conducted, but no treatment was identified for mortality. The overall scientific harvest of these trials was described in an article in Science as being thin. No trial reached conclusive results apart from the limitations of the chosen design of some of these trials. The trials were conducted late in the pandemic, and you can see from this figure, this gives three epidemic curves in three countries: Guinea, Liberia, and Sierra Leone. The first case was identified back in the middle of March 2014, and by August 2014, July, August 2014, the WHO have declared the epidemic as a public health emergency of international concern. But as you can see, the trials which were conducted, which are marked largely on the right here, they started to be conducted at the later stage when the epidemic was waning. And so there were insufficient cases to randomize in some of these trials. So the lesson one learns from that is that one needs to develop interventions very quickly during a pandemic. Okay, randomized controlled trials provide the most reliable evidence for the effects of medical interventions is beyond dispute. We will all agree on that. However, the traditional way of designing and conducting randomized controlled trials is slow and costly, and thus is not well suited for developing intervention during a public health emergency, where the speed and efficiencies are of essence. Even during peacetime, without pandemics, the need for improved efficiency in the design and conduct of clinical trials is well recognized. The slow pace of the traditional approach for testing new treatments stems from the fact that investigational agents are tested separately and each of the investigational agents involves separate phases. From after phase 1, you've got standalone phase 2 trials, and then preparations and conduct of separate phase 3 trials, and the whole process is repeated with each investigational agent. MAM's design, on the other hand, these designs were first developed by colleagues at the MRC CTU, and they were implemented in the STAMPEDE trial in high-risk prostate cancer. And the main features are as follows. First, we have a master protocol, a platform with a master protocol for assessing not just one treatment, but several treatments concurrently. And multiple treatments are compared to concurrently randomize patients in a combined control arm. And that is the main gain of efficiency. Because when you conduct separate randomized trials, each trial will have its own control group, and therefore you need more patients. There are also multiple stages of them to test for early futility. We only take products forward if they show some promise. So a shared control group means we need a smaller sample size per comparison and shorter time to identifying effective treatment. And the multiple stages allow for effective treatments to be dropped early, and the focus remains on the promising treatments. The platform aspects of the design also provides a shared infrastructure for conducting more than one trial at the same time. So those were the advantages. The challenges in MAMS are largely operational. The randomization is more complex to implement. For a start, the allocation ratio between the treatment and control is dynamic and changes depending on how many products are being assessed at the time when the patient is randomized. Other operational issues are summarized in a nice article by Sides et al. In Trials in 2019. One other issue also, which is highlighted, is that if one assessment of one investigational agent is completed, while there are some other investigational agents still being assessed, there is an issue when you publish the results of the completed trial, is that you will be unblinding the control group, the results in the control group, and and some of those patients in the control group will be shared by other agents which are still in the follow-up. So several of the major trials network during the current pandemic of COVID-19 used similar designs. That LISA used a shared control group in a platform trial. But I think only 2 of those trials used the full multi-stage MAMS design. And these are Active2 and Active3, 2 large platforms funded by the NIH in the US. And I shall focus the demonstration here on Active3 and its use of the multi-stage, multi-arm design. This is just description of how the network involved in this trial. The lead network in conducting the Active3 or the TICO trial is the INSIGHT network, which started initially as an HIV treatment trial, but adapted to also test treatments in influenza and more recently in COVID-19. The network is international, involved more than 300 sites worldwide, and is coordinated through 4 international coordinating centers in Washington, Copenhagen, London, and Sydney. The TICO trial was, although the lead network is inside, it was really conducted by a collaboration of 4 networks. 3 other networks funded also by NIH were coordinating this trial with INSIGHT. The design of TICO is a phase 3, two-stage MAMS design. The agents are evaluated as if they are in separate trials. So randomization, eligible patients are randomized to the investigational agents or to the matching placebo. But the allocation ratio between the active investigational agent and the matching placebo is dynamic, and it depends on how many other agents are being assessed concurrently. The idea is that at the end, for each investigational agent, one ends up with a one-to-one randomization between the active and the control. But because more than one investigational agent will be sharing the control group with others, the allocation ratio has to be worked out depending on what's being assessed concurrently. This is one of the operational complications of conducting MAMS. Investigation agents are assessed for safety and early signs of efficacy 5 days after randomization, and this is the first stage, so that only investigation agents that pass this assessment can move on to stage 2. So in TICO randomization, there are two disease severity strata. I don't need to go through the details, but essentially stratum 1 is less severe disease, and stratum 2 the severe disease. The population, by the way, in TICO are hospitalized patients with COVID-19, but with symptoms no more than 12 days. And for stage 1, the randomization is limited to disease severity stage stratum 1, which excludes patients who are sicker. For example, patients who have any cardiovascular disease as listed here, or renal disease, they will not be in stage 1, and therefore they're not eligible to be enrolled in the first stage. But they can be enrolled once an investigation agent has passed the first stage and moved to the second stage. So here is a pictorial, just picture of what happened. Stage 1, limited to disease stratum 1, and the outcome of that, after 300 patients have completed a follow-up of 5 days, is assessed by the DSMB, and if the product has shown signs of efficacy and a good safety profile, it can move on to stage 2. Otherwise, it's stopped at stage 1. The primary outcome for assessing these agents is what is called sustained recovery. And sustained recovery is defined as a patient is discharged from hospital and remains at home for at least 14 days without any need of rehospitalization. Sample size was calculated to require about 843 sustained recovery events to detect a difference of 25% in the recovery rate ratio. Stage 1, at the stage 1, another outcome is used, which is an ordinal outcome of 7 categories, ranging from the best category, which means that essentially the patient is out of hospital with no limitations on the usual activities, and the worst outcome is death. In MAMS trials, often in the early stage, the futility stage, we don't necessarily use the same outcome. In fact, this is an advantage. that in the first stage, some intermediate outcomes are used to gain efficiency. And but it is important that the intermediate outcome, which is used in screening products before they go to stage 2, that the outcome used in assessing them is correlated with the ultimate outcome, which is in this trial happened to be sustained recovery. But the intermediate outcome is the clinical status of patients described by an ordered categorical variable, as I said, ranging from the best category, which means no limiting symptoms, up to the worst category, which is death. And this slide here shows the concordance between the intermediate outcome used in stage 1 and the ultimate outcome, which is used in stage 2. And this is done for the first product assessed in TICO. It's neutralizing monoclonal antibody manufactured by Lilly. And you can see that for this product, there is no difference between the two treatment groups for the intermediate endpoint, as you can see on the left, and ultimately also when the trial was concluded or stopped at the end, and you can see that the same result was obtained for the sustained recovery outcome. So the requirement for the intermediate outcome used in stage 1 in the MAMS design is that they be correlated with the main outcome, primary outcome with the study. They're not necessarily surrogates, but at least they have good correlation. And this demonstrated this correlation at least for the 2 outcomes used in TICO. So the criteria for moving an agent from stage 1 to stage 2 is very liberal. And the idea is that you don't want to stop a trial for one agent unless there is fairly good evidence that it's not going to be successful at the end. And that's why it uses a one-sided p-value of 0.03. 300 patients are required and to be able to detect an odds ratio of 1.6 in terms of the intermediate outcome of the ordinal outcome for to assess products in the first stage. And you can see that this requirement is really not very strict at all, using a p-value of 0.3. But the choice of this value was demonstrated by Folman and Proshen, that to have a minimal impact on the overall power. Originally, the study was in terms of the primary endpoint, was powered for to provide 90% power to detect a difference of a 25% increase in the rate of sustained recovery, and by using the intermediate outcome and this criteria for moving product forward, the power is not affected at the end. But of course, it has the added advantage that more treatment are evaluated. And that paper by Folman and Proshen also demonstrated that if of all the investigational agents, of all the treatment that have been affected, if half of them, for example, are efficacious, then 40%, then more than 40% will be identified. Sorry about this. So here are the investigational agents that have been assessed in TICO. So far, there were 6 agents. 3 of them have already been stopped, and 3 are continuing. The three which are continuing are neutralizing monoclonal antibody by AstraZeneca, and then two other molecular partner and a Pfizer protease inhibitor. These are still conducted. The 33 were all stopped at stage 1 because they did not pass the first hurdle of showing some signs of efficacy. So in the remainder, I will just have to, I would like to share some thoughts about the design and implementation of the vaccine trials conducted for COVID-19. The FDA guidance for the vaccines recommends a 1-to-1 allocation ratio for the randomization between the vaccine and its matching placebo. The follow-up is ideally 1 to 2.5 years to assess the duration of protection and potentially to assess vaccine-mediated disease. At the minimum, there is a requirement of at least 2 months of follow-up after the last dose for safety. And the point efficacy for success, the point estimate for the vaccine efficacy is to be more than 50% with a lower confidence bound of more than 30%, because you don't want just to prove that the vaccine is significantly better than the placebo. You want to show that it's really substantially better, and that's why you have to exclude an efficacy of 30% or less. As we all know, I mean, the vaccine story for COVID-19 has been a huge success. There are more than 5 now who have been approved, and they have very high, at least in the short term, very high vaccine efficacy. We're talking about vaccine efficacy over 90% for most of them. Typically, the primary endpoint for these trials have been virologically confirmed COVID-19, and a typical sample size is about 30,000 to about 40,000. Median follow-up is about 2 months, and the methodology by which vaccine efficacy is estimated or the outcome is estimated uses either a Poisson model or a Cox regression model. So here I'll just go through very quickly through some of the issues in the design of these trials. First, one of the major issues, obviously, is the impact of the availability of an effective vaccine on an ongoing placebo-controlled trial of another vaccine. I mean, and the other 2 points, obviously, are going to be the other 2 points are just optimal allocation and how do we estimate from a short period the duration of protection. Now, in terms of volunteers in the placebo group, obviously, one cannot, during a pandemic, if there is an effective vaccine, they should be allowed to use that effective vaccine. And this can be done either by a crossover design, where they are, after a certain time, they are unblinded and being allocated to the active treatment within the trial itself. But also, in a lot of these trials, while they were ongoing, there were some other vaccines who have been licensed, and some of the people allocated to the placebo group would have been eligible to receive these vaccines, and therefore they will be unblinded and given an active vaccine. So how did these trials cope with the fact that in the control arm, people are going to receive active vaccine? Well, essentially, the follow-up was censored at the time when, of each patient, censored at the time when they were unblinded and they were receiving an active vaccine. However, this raises an issue. censoring at the time of unblinding have lead to a potential bias in estimating the vaccine efficacy. And this is particularly the case under a Poisson model. The reason for that is that when the placebo group is censored, they have shorter follow-up. And therefore, most of their follow-up is going to be during the early time of follow-up during the trial, while the active group is followed for much longer. And if the infection rate changes over time, and if, for example, it was high at the beginning, and then it was much later infection rate at the end, we'll not be comparing like with like, and therefore that will introduce a bias in comparing the active vaccine group with the placebo group. And the way out of that is obviously to use a Cox model rather than a Poisson model, because a Poisson model assumes you have a constant infection rate during follow-up, and that is clearly not the case. And then we can also adjust for calendar time in the analysis. All this assumes that the censoring itself, because of unblinding, does not depend on the risk of infection. But if it did, then one might have to use what we call inverse probability of censoring weighting to adjust for any potential association between censoring and the infection and the risk of infection. Now, the next point is assessing duration of protection. And there is a nice paper by Follman and others, and also by Lin et al., which they propose using using the data collected from trials who followed patients even after they have announced their results and proved that the vaccine was efficacious. They continued to follow patients after they crossed over from the placebo into the active treatment. And the best way to analyze that, obviously, is to use a Cox model, but use calendar time at the analysis time, not the follow-up of individual patients. What about the optimal allocation ratio in these trials? As I said, the FDA recommended a one-to-one randomization, which is obviously simpler, but it's not optimal in terms of minimizing the target number of cases, which is needed. The minimum target number of cases actually will depend on the vaccine efficacy and the alternative hypothesis. So one can show that, for example, for a vaccine efficacy of about 60%, the minimum number of cases required to achieve the required power of a 90% power to exclude an efficacy of less than 30% will be approximately 2-to-1. But then there is a trade-off between simplicity of having a one-to-one randomization and efficiency in terms of reducing the number of required cases. However, it's not clear whether optimality should really be defined in terms of a minimum number of cases required, or actually the shortest time to complete the trial. And these two things are not the same. So a 2-to-1 randomization has advantages. More vaccinees, other advantages, obviously, more vaccinees will be receiving the active treatment. Sorry. and therefore it will be more attractive for trial participants. So in summary, in global pandemics, the speedy evaluation of therapeutic and preventive intervention is really paramount. Speed is the essential thing, as we have seen that one of the major reasons why the trials conducted during the Ebola outbreak were not very successful. An adaptive platform trial with MUM designs are efficient and can be swiftly implemented during a public health emergency, as has been demonstrated by a number of trials conducted during COVID-19. Efficient trial design obviously is not enough, but one would need also a global collaboration to be able to conduct these trials in good time. So finally, I just want to acknowledge the Active3 and TICO study group and my colleagues, Max Palmer and others at the MRC CTU who helped me with these slides. Thank you very much.